Part 8 of our videos on evolution has to deal with phylogeny. The concept behind this is that phylogenetic trees and cladograms are graphical representations or models of evolutionary history that can be tested. Cladograms and phylogenies are diagrams, essentially, that group items together based on the number of common characteristics. Um, to do these in general, you start with determining the number of shared characteristics that an organism's exhibit, and then you arrange those items in a tree showing the most common possible, the most commonality possible. One note, a lot of times students can get tripped up on the difference between a cladogram and a phylogenetic tree, and in general, for our purposes, there really isn't too much different. People tend to use those terms interchangeably, and actually there is some disagreement amongst the evolutionary scientists as to what the actual difference is. So for our purposes, we're going to use them relatively interchangeably. So phylogenetic tree, there's a couple of different ways that you can group the data. You can look at the shared derived characters, and these are physical traits that represent evolutionary history. The other sort of term for that would be a homologous structure. But you can also group them by the DNA or protein sequence data, looking at the differences in sequences that would accumulate as species evolve from one to another. Here is an example of a phylogenetic tree that's using shared characteristics to group them. So essentially, you have at the very bottom vertebrates, and so everything above that would have a vertebrate, and then at some point there was a branching in the tree where you have bony skeleton organisms going one way and sharks going the other, and then a further break for four limbs, so that left our poor ray-finned fish behind, and etc. on up until you have all of these different characteristics, some of which organisms share and some of which they don't. Of course, can be way more complicated. We can look at the entire phylogeny for vertebrates and see that it is quite complex. Here is a complete phylogeny. So we've got bacteria, archaea, vertebrates, all of that is in here. So it is quite amazing. One just sort of note about looking at phylogeny, it's a lot like reading a family tree. The root of the tree represents the ancestral lineage, and the tips of the branches represent the descendants of that ancestor. As you move from the root to tips, you are moving forward in time. When lineage splits, we would call that speciation. It's represented as a branch on a phylogeny. When a speciation event occurs, a single ancestral lineage gives rise to two or more daughter lineages. So you can see in this picture here for number 2, you've got the ancestral lineage coming up, and then there's a speciation event which has occurred, and now we have 2 daughter lineages there. Phylogenies trace patterns of shared ancestry between lineages. Each lineage, I wish that word didn't have to be said so much, but has a part of its history that is unique to it alone and parts that are shared with the others. So you have this shared history of B and C coming up, but then when there's the speciation event between B and C, now they have their own unique histories. Similarly, each lineage has ancestors that are unique to that lineage and ancestors that are shared with other lineages, and we would call these common ancestors. So if we were to look at the right at the branching point between A, B, and C, this first one, that before that would be a common ancestor of all 3, but after A has branched away, then we're having common ancestors of B and C, and so on. If we were constructing a phylogenetic tree, the most typical way that you would see it is being given a table of shared characteristics and then needing to organize them in a tree diagram showing the simplest possible evolution. And this uses the concept of maximum parsimony, which is all else being equal, a trait is assumed to evolve once it's present in all the descendants. So if you were going to make a table with this data here, you would see the chimpanzee has all of these characteristics: opposable thumb, four-chambered heart, amniotic egg, lungs, and a spinal column. Well, if you look at the bottom of the table, the lamprey has none of those characteristics. So you could use this data to make a phylogenetic tree, and if you would like to try that, you can pause it and you can show me in class. Just one note to remember, trees are hypotheses, right? We don't say that these are exact. It can be continually revised as more data is gathered, as we have better ways to look at the data. The relationships between organisms can be revisited. There's also the role of computers, because this is quite complex, and so computer analysis is needed to determine the numbers of similarities when we're talking about entire DNA protein sequence information.